Thank you and welcome to this four-part series on precision medicine, moving theory into practice. We are very excited about the discussions to date where we've talked about how we leverage precision medicine for patient needs. How do we really think about measuring real-world effectiveness and value? And today, we're going to be discussing using precision medicine in clinical practice and both the impediments and the opportunities to doing so. And then next week, we will be looking at effective payment and reimbursement models. So I want to begin just by noting that our previous webinars have been recorded and are available on the Schaefer Center's website, or they will be once the series is complete. And I also want to thank the Agency for Healthcare Research and Quality for their support of this webinar series. Nothing you hear, see, or that we do reflects official policies of HHS or the US government. So let me begin by introducing our first speaker, Dr. Jennifer Malin. She is the Senior Medical Director for Oncology and Genetics at United Healthcare. And in that role, she provides clinical and strategic leadership for improving the health and outcomes of cancer and genomic medicine for the United Healthcare membership, which is one of the largest in the country, if not the largest. She's also a prolific scholar. She's authored more than 100 peer-reviewed papers and is widely recognized for research on the quality of cancer care. And she's done quite a bit in this field to think about how we can get patients better care. So Jen, welcome.
Thank you, Dana, and everyone for hosting this event, at the Schaefer Center for hosting and for inviting me to speak here today. I'd like to start just with a definition of precision medicine. The term gets used in a variety of different ways, but this is the definition from the Oxford English Dictionary, which I think is a good way of grounding our conversation today. So precision medicine is medical care designed to optimize efficiency or therapeutic benefit for particular groups of patients, especially by using genetic or molecular profiling. And so the conversation today, I think, is really focused on this idea of precision medicine using genetic or molecular profiling. And, you know, there's a reason that this has become such a focus of attention recently, and that's because the cost of sequencing DNA has decreased dramatically. So if you look back in 2001, when the first human genome was sequenced, it was around $100 million to sequence the genome at that time. And now, you know, certainly for research purposes, you can sequence an entire genome for around $1,000. However, you know, when you're looking at sequencing genes clinically, there's not just the cost of the sequencing itself that you have to consider, but the cost of interpreting the data, which today still remains a fairly manual process involving, you know, experts in genetics and understanding when you see a change in the DNA a variant. Is that a clinically significant variant? Is it a mutation that could be pathogenic, or is it just a normal variant? And so that cost increases the more genes you sequence. And so that's an important consideration as you're thinking about, you know, the cost effectiveness and in applying this technology in new situations. So when is a genetic test ready for clinical use? And this is the way we think about it as a payer, although I think it's also, you know, really the way, at least I think about it as a clinician as well. So to have clinical utility, a genetic test must lead to an action that's been shown to improve patient outcomes. And so that's, I think, really critical. So we see, a variety of uses of genetic tests. The broadest base, and I think where we often see the greatest excitement is really the area where there's the least amount of evidence of clinical utility today, and that's predictive testing. And this is when a test is used in someone who's asymptomatic to try to determine if they have a disease or are at risk of a disease. And so for predictive testing to have clinical utility, it needs to lead to a change in clinical management that improves outcomes of the individual or their family members, or providing closure by ending a diagnostic odyssey. And for diagnostic testing, which is kind of the next level up, you need to lead to a change in clinical management, again, that improves the health outcome for that individual. So it doesn't help to make a diagnosis of a horrible, debilitating neurological condition if you can't do anything to improve their quality of life, improve their prognosis, unless it ends a diagnostic odyssey. And then the area I think that we're most focused on today in terms of the idea of precision medicine is really this targeting of therapy. And that's really where there's the most evidence today for precision medicine and genetic testing, and using genetics to better target therapy. Where do we see precision medicine in the clinic today? So as I mentioned, this is where the most evidence of clinical utility is. Targeted therapies are the standard of care for many, if not most, cancers today. There's still an important role for standard chemotherapies and biologic therapies, and new checkpoint inhibitors, but there are now over 100 targeted therapies for cancer across the different types. Increasingly, we're starting to see gene therapies that target germline mutations. So most of the cancer therapies target mutations that happen in the cancer cells that we call somatic mutations. Gene therapies target mutations that are causing inherited disorders. So we see therapies like Luxturna and Zolgensma for blindness, in Luxturna's case, caused by a mutation in a gene that affects the eyesight, Zolgensma for a muscular dystrophy inherited condition. But there are therapies in development for sickle cell disease, hemophilia, thalassemia, that will be potentially life-changing for these inherited disorders. It may seem kind of, circular, but in this targeting therapy area for testing to have clinical utility, there needs to be a therapy available. And then here's one of the areas where I think has caused kind of the greatest tension, perhaps, in terms of payer coverage policies and access in the clinic, and what I would argue maybe unintended consequences that have ended up limiting access. So because it's now so, the costs have come down to sequence the genes, we see these very large cancer panels that have over 300 genes, where most of those genes don't have clinical utility because there's no therapy available today. The cancer that has the most targeted therapy options currently is lung cancer. And even for lung cancer, NCCN guidelines only recommends testing for 7 biomarkers that have a specific therapy associated with them. Although they caveat this by saying that it should be part of quote, broad molecular profiling. And I'm going to show you some data on how this leads to unintended consequences of really the healthcare system not really having a clear idea of where to put this in clinical practice. And I'll just kind of end with this. It's that generally when a physician goes to prescribe a treatment now for, if the therapy is targeted, the health plan is going to require evidence that the mutation or the biomarker is present prior to covering that therapy. So as I mentioned, guidelines emphasize, potentially getting these very large panels so that, you might be able, the argument is you might be able to identify people for clinical trials. But what we see happening in practice is that most patients don't even get the bare minimum testing. And so this is out of our prior authorization platform for drug therapy. And you can see here these dark blue bars and this is for lung cancer. These dark blue bars represent where the mutation status of the patient is unknown. And so you can see, even for the most common mutations, EGFR and ALK, their mutation status is unknown. And all of these are recommended to be tested for by NCCN guidelines, and we can see most patients are not getting tested. And so what my concern is that because the guidelines are maybe somewhat vague and are seeming to advocate for very large panels, most people are not getting tested. And what we really need is a focused effort to say, okay, for this set of genes, some of the PD-L1 is not a gene as a biomarker, but for this small set, can we get consensus and just have pathologists order them routinely on these patients, so that by the time the patient makes it to their oncologist, they have the information available for them.
Thank you. Let me jump in here, and that's very interesting data, and we'll come back to it in the discussion. But first, I want to turn to John, who is a professor and chair for the Department of Translational Genomics and director of the Institute for Translational Genomics at our own Keck School of Medicine. His expertise spans a broad range of research disciplines, including germline genetics, tumor profiling, cancer cell biology, functional genomics, and health disparities. And in 2021, he was inducted into the AACR Fellows of the Academy. Congratulations. He also currently serves as a member of the NCI Board of Scientific Counselors and as a member of the Scientific Advisory Board for Breakthrough Cancer. Welcome, John, please.
Yeah, thanks. And it's really wonderful overview by Jennifer. And I think there will be some overlap and redundancy in our presentations, but I think provide a few additional examples, and I think some additional perspective as we sort of think about precision medicine. What is it, what it means, and some maybe unique ways as we think about applying this approach. And just thinking about precision medicine and, as this sort of approach to, to transform healthcare in the 21st century, and building these engines to enable medicine and empower change through training, application, and practical implementation of this approach within a major academic healthcare system. And this was, sort of developed by our group as we were recruited here to USC 4 or 5 years ago, in terms of what we see our impact will be as we move forward. Jennifer mentioned the definition, and a number of from the specific definition from one of our dictionaries, and the NIH also sort of provides the National Institutes of Health provide a definition for precision medicine, as well as an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. And so this again expands, I think, the overall sort of architecture as we think about precision medicine, away from just the specific or unique genomic profile that an individual may have, but also considering again environment and lifestyle as well. And it will definitely involve merging and integrating across advanced molecular technologies, but also together with current clinical tools to personalize health through individualized prevention, monitoring, diagnosis, and intervention. And with that, I tend to look at precision medicine as sort of a pie as we think about precision prevention, precision diagnostics, precision therapy. And I'm actually working on a fourth slice that will be precision palliative care and survivorship, because after patients are treated, basically we still have to consider their life after or while they're still suffering from a chronic disease or disease, and some of the very unique and individualized aspects we can bring to bear and take into account to improve their overall quality of life. And again, as we think about precision and prevention, can we actually prevent a disease? And I'm going to focus a little bit on cancer today. from occurring in the first place. So through understanding, epidemiology, population science, and modifiable risk factors such as diet and lifestyle, and genetic risk. And of course, at some point also, as we work towards integrating the social determinants of health as well. And then there's detection. So, at presentation, can we detect early, accurately, and non-invasively? So what are our best approaches to actually detect a disease or a condition as early as possible? Because that may allow us to have a better and more effective approach towards managing or treating that particular ailment. And then finally, intervening. So once something is diagnosed, right, how do we best and most appropriately intervene, again, based on an individual's more personal and individualized profiles. And again, as Jennifer was mentioning, identifying the right treatment for the right diagnosis. Many of the core components, I think, as we think, about the various components that feed into precision medicine, the clinical professionals, the nursing and research coordinators, the clinical geneticists and molecular pathology, the genome scientists and molecular technologies, medical technology and engineering, as we consider imaging, right, as also part of the precision medicine architecture, biomedical informatics and biocomputing, which is really big, as we consider we're being in the era of big data. And I'd be remiss to not consider the importance of social work and patient navigation. Again, identifying those really unique and individual situations that individuals live in, right, to best tailor approaches for mitigating and managing these diseases and ailments. And again, just to try to keep this simple, just focusing a bit on diagnostics and therapy, because these two things definitely go together. So we think about, as Jennifer was mentioning, targeted therapies. In many cases, there's what we call a companion diagnostic, right? Something that tells us that this is a specific patient that might benefit better from a very specific treatment approach. She mentioned that greater than 100 targeted therapies out there, and importantly, many of these in cancer. Again, I'm focusing on cancer. A number of these targeted therapies are tied to very specific and unique molecular alterations that are present in either specific tumor types or across various tumor types. And I think in cancer, the poster child was the BCR-ABL translocation in leukemia, and Charles Sawyer's and their work in identifying or developing imatinib and dasatinib as a very specific, Brian Drucker and Charles Sawyer developing imatinib, which is also known as Gleevec, towards the treatment of leukemias that harbor this event. But we've seen a considerable growth in this area, and also, as mentioned, the recent development and advent of utilization of immunotherapies, PD-1, PD-L1, immune checkpoint inhibitors for the treatment of various forms of cancer, including melanoma, lung cancer, and other cancers, nivolumab and pembrolizumab, which are drugs that are now used in the treatment for various forms of cancers that overexpress these immune checkpoints, by where our immune system is given the opportunity to help fight off the cancers. So instead of patients going on broad-based chemotherapies, of a specific molecular alteration is identified in that cancer, the doctors can then choose from a series of specific targeted therapeutics. And one of the poster childs, I mentioned Gleevec, but the other one is vemurafenib, an inhibitor of a very specific mutated form of a protein in melanoma. The V600E mutation was identified through cancer genome sequencing. Mike Stratton and the folks at the Sanger Center in the UK identified these mutations in melanomas, which led companies to identify very specific small molecules that targeted specifically the mutated form of that oncogene. And by turning that oncogene off, you could go from scans where patients just have considerable cancer burden to seeing those tumors essentially melt away over time. Again, many of these patients would have originally gone on chemotherapy, but because we can identify these mutations, we can provide these targeted therapies that are very, very effective. And of course, in many cases, these responses are transient, as the cancer cells eventually figure out how to evade even these targeted therapies. Now, what's important, I think, as we consider precision medicine are the possible confounders. We think about cancer disparities, or those cancers that disproportionately affect underrepresented minority individuals. As we think about cancer incidence rates and death rates and differences, meaning this high rate ratio, meaning that these cancers disproportionately are represented or have higher burden in Blacks versus whites. And this is stratified by female and male, and also as we consider death rates. And we know that there's a combination of factors that influence these what we call cancer health disparities, where there's a disparity or a higher burden in one racial or ethnic group versus other racial and ethnic groups. We know that societal factors such as race and social determinants of health play a huge role in those differences, but we also more recently are beginning to uncover potential biological factors related to underlying genetic ancestry that also play a role. And we know that these two components interact towards the impact on the host and the tumor biology. However, there's been a significant dearth in the information or the data that we have on tumors that are derived from individuals from underrepresented racial and ethnic groups. And even in large studies such as the Cancer Genome Atlas, where over 11,000 tumors across 33 different tumor types were sequenced and profiled to identify the common mutations that occur in these cancers. However, overall, less than 10% of these tumors were derived from individuals from underrepresented minority groups such as African Americans, Hispanic Latinos, or Asian Pacific Islanders, or Native Americans, even for cancers that disproportionately affect these groups, such as prostate cancer, triple negative breast cancer, colorectal cancer, endometrial, leukemia, and multiple myeloma. And there have been a number of studies that show that there are differences. If you look at genetic ancestry and race and stratify mutation profiles, we see significant differences, even in pathways where we have targeted therapies, such as the PI3 kinase AKT pathway. And why is this important? Because in many cases, as we think about precision oncology and precision treatment, many of the data that we're using to identify these mutations that then lead to novel therapies are discovered in largely, for instance, white or European descent patients. So this was a component of a document that actually was commissioned by Congress, which was a progress report on cancer health disparities, and myself and Shanita Hughes-Halbert, who were members of the steering committee, wrote this section on the imprecision of precision medicine. You know, as we apply precision medicine in the clinic, particularly as we consider precision oncology, the limited knowledge we have on the cancer biology from tumors derived from racial and ethnic minorities diminishes the potential of precision medicine in these populations. And so we want to, get towards a broader view and a broader understanding of the overall molecular context of cancer across different groups and populations, so that we can be more precise, even more precise in these approaches for precision medicine. And I end with this slide, and that even when we have a great biomarker, a great target, and a great therapy, right? There can even be disparities in the application. So one of the early applications of precision medicine was the identification of the HER2 oncogene in a subset of breast cancer patients, and the development of a very specific drug, trastuzumab, that's used to treat those cancers. However, scientists at North Carolina published a paper back in 2016, and there have been a series of these papers that published that show that there are disparities in the utilization of the test and disparities in the utilization of that treatment. Even though we have a great model for precision medicine for this particular form of breast cancer, we can even see disparities in the adoption of this approach, meaning certain groups might get the test and certain groups might get the targeted therapy compared to other racial and ethnic minority groups. So we have to be very, very careful as we consider precision medicine and its application to ensure that these types of healthcare innovation are available to all people. So I'll stop there and just finish up with, we know that there's tremendous promise with precision medicine, but there are also a number of challenges that we have yet to overcome to see broad application of this approach in the treatment of patients with various diseases and conditions. So I'll stop there. Thank you.
Thank you both. And I want to come back to this issue of disparities. But first, you mentioned precision diagnostics and precision therapeutics, and I think both of those are key. But both of you discussed the fact, and this is about clinical translation. And John, you articulated that we see these disparities by race and ethnicity as well. And Jen, your data show that when people aren't always getting tested, and as John said, they're not always getting treated. But yet at United, you've led a lot of efforts to do evidence-based care to come up with, and in fact, you may be at this point, United Health probably delivers a lot of care in the United States. How do we get widespread adoption? What are the efforts that are underway on the insurer side to make sure that everyone has access?
No, I mean, these are really important questions. And I want to circle back before I get to it, just to underscore the issue of disparities and the concerns that you know, the world of genomics can potentially make that worse if we're not very focused on it. There was a recent study that came out that showed that one of the tests that's used widely to help figure out which women should get adjuvant chemotherapy for breast cancer. And this group of researchers evaluated the outcomes in women who are African American. And it turned out that the kind of general low risk group, if you looked just at the low risk, you know, the low risk threshold where you wouldn't normally give chemotherapy to women who are of African American descent, their risk of the cancer coming back was double the, you know, the overall population, which was predominantly white. And so it's very, very important, I think, you know, on this implementation continuum, that we get that last mile implemented, but it's probably equally, if not more important, that we make sure we have real diversity in our research studies and in the way we develop these diagnostic tests to guide therapy in the first place. Because if they're not accurate, it doesn't really help to get it, you know, to get it to the patients in need if they're not accurate in all the different patient subgroups. So, you know, but in terms of like what we're doing today at United, I mean, we have a real focus on, first of all, you know, making sure that, you know, people do have the test, you know, before we prescribe a therapy, that the test is done. We're looking to, we have a pathways program that we've rolled out nationally, where we have basically it's a pay for performance program or a quality initiative, where we provide an incentive for oncologists to follow our pathways. We haven't as yet required the molecular testing to be on pathway. It's a little bit of a nuanced discussion, because sometimes it is hard to get the lung cancer tissue. But I think, you know, increasingly, I think we're moving into a world where we may start to say we're going to require that to be considered on pathway. Because as you can see, too many people are just not getting it done. And then, you know, through forums like this and other different working sessions, ASCO's having one later this year. There have been ones that have been held by, you know, the, you know, lung cancer advocacy groups, really trying to figure out, can we do what we did in breast cancer years ago and create a path where pathologists routinely order the set, you know, the basic set of tests that everyone needs, so that it's available for everyone, and kind of working through those systems issues, which, you know, it's frustrating that it's not easier than it is, but those system issues are really the things that need to be, you know, to have something implemented that's just routine and doesn't require someone to go back and reorder the test after the pathologist has already, you know, read out the slides. what should help, I think, make things more smooth.
That's it. Thank you. That's very interesting. John, did you want to add to that?
Well, not too much. I think, the one, I think, aspect is, we are working towards development of hopefully more improved technologies that might have even more sensitivity, specificity to be able to detect these mutations on the slide themselves, or utilizing approaches such as circulating cell-free DNA profiling, or liquid biopsies, where we don't need the actual tissue to identify these potential therapeutic related or targeted therapy related biomarkers. So I think, within the next 5 to 10 years, we'll see even more growth. So, we'll hopefully cross that hurdle where, being able to detect won't be the challenge. The challenge will be adoption, and who gets it and who doesn't.
Right. And so, and how much of I want to keep on this theme just because it shows the clinical translation part. How much of this is due to just who has access to academic medical centers and places where historically they've been conducting research versus community settings? Is it part of it that just the knowledge base isn't there on the provider side in some of these areas?
I think, through venues like NCCN guidelines, and I think the information transfer happens very rapidly. Mm-hmm. And even things like, like insurance companies, which are often viewed as the barriers, our entire coverage system for drugs is linked to NCCN guidelines. So we update our platform as quickly as NCCN guidelines get updated. So there's pretty rapid information dissemination. And I think because insurance companies rely on NCCN so heavily, that's caused them to even update their guidelines more quickly, right? So it creates a circle where everyone's using and we're creating real-time platforms. The next system is to have actually these platforms integrated with the EHRs. So I think that information is getting out there at the point of care. The challenge is just the coordination of care. And so you have to get, it's a radiologist, or, I'm focusing on the lung cancer example, but, you could name your example, but, it's a radiologist or a pulmonologist who's doing the biopsy, who sends it to the pathologist. The pathologist will says, well, at the time I get the biopsy, I don't know if the patient has metastatic cancer or early stage cancer. Someone has to tell me that they have metastatic cancer, because if I order it in someone with early stage cancer, I'm not going to get reimbursed. And so then you say, well, isn't there a straightforward way, like that there can be a box that someone checks on the form that says metastatic so they always know? And then because that's not there, then the patient gets to the oncologist, and the oncologist says, okay, we can tell you have lung cancer, but we still need to order these other tests. And so I'm going to order these other tests to figure out which treatment to start you on. And so this person at this point has waited like 3 weeks to get into the oncologist. And they're like, I don't want to wait anymore. This cancer is spreading throughout my body. Start something right now. And they don't want to wait. And then, or, you call up the pathologist and they say, well, I have enough tissue to do one more test, but I don't have enough tissue left to do all 7. So which one do you want to get? Or do you want to go back and get another lung biopsy? And the patient, of course, doesn't want to go get another lung biopsy at that point. So it's this complex system of, processes that, like, you kind of like scratch your head and you think, like, really, this is what's making it so hard? But that's what makes it so hard.
It's not just precision medicine. It's everywhere in healthcare, for sure.
Yeah.
Exactly.
So one of the questions that you spoke, John, about a lot of the original research, for example, on the human genome, ironically, was done on very homogeneous populations, European descent. That's
it was like 2 people.
Yeah, 2 people, right? And I remember we had a presentation at the Schaefer Center Advisory Board from Ann Wojcicki, who runs 23andMe. And, you know, if you looked at their original database, you'd think that 20% of the population was Ashkenazi Jewish descent because that was who was applying, you know, sending their DNA because they were curious about their heritage and also their inherited disorders. So how important is it that we get broad representativeness into these, as both of you were articulating, actually, to make sure that we get the right treatment to the right patient?
No, it's critically important, you know, as we consider this whole concept of diversifying cohorts in biomedical research and diversifying clinical trials, right? Because we're talking about, you know, the genomic profiling and molecular profiling, but clinical trials is another, you know, huge issue in terms of, you know, lack of diversity where drugs are approved based on data from, you know, large, you know, definitive phase 3 clinical trials that are largely European, maybe 97% European descent. And, you know, a lot of clinical trials are beginning to be conducted offshore in other countries that also have, you know, very homogeneous populations. And so yeah, I mean, it's really important. And I think there is mounting evidence probably over the last 10 years that, you know, if we consider the molecular profiles of cancers and probably even the actual biology of these cancers, right, that there are unique features that have to be considered. And it's not just in research. You know, for instance, a good example are the gene expression profile clinical tests that are available for predicting prostate cancer recurrence. Prolaris, Decipher, these are clinical tests that, you know, a pathologist or doc can send a biopsy sample to the company. They perform a gene expression test to determine what's the likelihood of that cancer, right, being a really bad cancer, right? And those tests were developed off of, you know, where the biomarker test was probably, you know, developed and validated, right, of large populations of prostate cancer cases. But, you know, many of those patient populations were like from places like the Mayo Clinic in Rochester, right? And there's just like no diversity. And however, prostate cancer is one of the most significant cancer disparities. So you have a test that's based on data from largely Europeans that's going to be applied to Black men. Now, why that's really important? Because a number of more recent studies where we have been able to look at the gene expression profiles of prostate tumors from Blacks versus whites show unique patterns, right? By where tumors from Blacks have an enrichment of pathways associated with inflammation, right? And many of those markers are not being included in these tests, right? So we have to get away from this overarching concept of generalization, right? We're generalizing, right, the results. We develop them in one place, in one setting, and then say they're going to generalize in every setting. And we know that that's probably not the case. So it's going to be critically important for us to continue to work towards diversifying these cohorts. And one example is the All of Us study. I'm sure many of you have heard of that. So the brainchild of Francis Collins and some of his colleagues to amass a cohort of 1 million individuals by where biospecimens will be collected, blood. And I know I went over and I consented, and the lady put 8 tubes on the table. Like, I'm like, you're not taking 8 tubes of blood from me today. I don't know what you're thinking, but they're trying to take, you know, blood samples.
Give me back the informed consent.
Give me back, no way, right? And to follow people, right, longitudinally, right, for decades, so that we can understand, you know, what are the lifestyle, epidemiological, environmental, and biological features that are associated with these diseases towards, you know, better disease prevention. And so, you know, most, I think as of last week, one of the principals from All of Us gave a talk, and I think they're at about 20% underrepresented minorities, which isn't bad. But we hope to see, you know, studies really focus on trying to advance diversification in biomedical research.
Wow, well, that's fascinating, but also quite troublesome that we might have to wait decades to catch up on here. I want to get to some of the audience questions. We're actually running out of time quickly. So, and one of the questions is about how close we are to kind of algorithmic diagnosis and prediction, and how much is there too much variation in outcomes, and the clinician really matters? And I'll let Jen start for this one. Can we replace doctors with a machine?
Yeah, so I would say
just oncologists. I don't want to get hate mail.
So the short answer is no, and I can just kind of go into the example of developing pathways. So, developing pathways, we look at the data from the clinical trials on efficacy and toxicity. We then look at real world evidence in terms of hospitalization rates, duration of therapy. And even though we put it all together in a very concrete format, right, with numbers, hard data, it takes experts to interpret it, right? Because trial A gets done with against drug B that was on the market, and then drug C comes out and is never tested against drug A because it was B versus C. So you have to understand all these things. Going back to lung cancer, if you were to just look at the lung cancer trials over the last, outcomes of the trials over the last 2 decades, or maybe 15 years, not 2 decades, you might think that the drugs that, some of the kind of standard chemotherapy regimens that were used back then are not as good today. But it's just because, our imaging and diagnostics have gotten so much better that we're picking up people earlier. And so just the average life expectancy, not, accounting for any therapies, is better than it was 15 years ago. And so you have to have, still today, and that's, in the context of reviewing data for pathways, but it's just as true, if not more so, at the bedside. You have to be able to assimilate all this information. And right now, well-trained physicians are still the best, most rapid computers at trying to assimilate all that data and put it in the context of that person sitting in front of you with the disease.
Okay, we're almost out of time. So John, last question to you, which is, you mentioned liquid biopsy and blood biomarkers. They really have the potential to revolutionize what we're doing, but also greatly expand the patient population. What do you think the future holds for that space?
Yeah, well, again, there are lots of different diseases and conditions, and I'm really focusing a little bit on what's going on in cancer space. So a company called Grail, disclosures, right? And they are developing a pan-cancer liquid biopsy test for early detection across 10 or 12 different tumor types, I believe, I'm going to say. And are advancing really quickly with very, very large trials of hundreds of thousands of patients around the world, and trying to pay some attention to diversification, right, to make sure that there's broad diversification so they can really understand the true impact of these tests, or effectiveness of these tests. And so I think 5 to 10 years from now, I believe they will become more and more routine. But again, at the end of the day, right, you've got you've got payers, you've got reimbursement. Are these tests going to be a part of standard, right? Because the hope is to catch these things early, right? I mean, so you want that test, right, before a cancer is really maybe even manifested or had the chance to take off. So. But I think, I have hope that, in the not too distant future, we'll begin to see more broad utilization and application of these types of tests in the clinic.
Great. Well, I want to thank you both for taking time, and you've hinted at also both, as you said, the promise and the pitfalls. Our next webinar next week, we'll talk about reimbursement. And obviously, if we want all this to proceed, we have to do it in the most cost-effective manner possible. So I want to thank both of you for your time. It's really stimulating, and take care, everyone.